Welcome. In this video we are going to discuss about transfection in animal cells. Now, direct transfer of the DNA in the animal cells can be done by two ways. Either you can force cells to take in the DNA by breaching their cell membrane, that is one way, or the other way is you can exploit the natural ability of the cell to internalize molecules from the environment. Either you can force the cell to take in the DNA by breaching its cell membrane, or you can exploit the natural ability of the cell to internalize molecules from the environment. By these two ways you can transfer your target DNA into animal cell. Now, the term transfection was originally coined to describe the introduction of phage DNA into bacterial cell. Similarly, forcing animal cell to take up DNA from the surrounding medium using various chemical method or physical method is also termed as transfection. Transfection is all about forcing animal cells to take up DNA from the surrounding medium using chemical method or physical method. So that is the reason why this transfection is a highly efficient way to introduce DNA transiently and stably into the cultured cells or the cells in vivo. It is a highly efficient way to introduce the DNA transiently and stably into the cultured cell or the cells in vivo. The term transformation is applied to animal cells, and the term transformation refers to a stable change in the genotype, and this stable change in the genotype is brought about by incorporation of the transfected DNA into the genome or its long-term episomal maintenance. But this transformation term is also used to describe oncogenic transformation. What is oncogenic transformation? It is the change in phenotype which occurs due to activation of oncogene. So the change in phenotype that is brought by activation of oncogene, this phenomenon is also described by this term transformation. Now, the fate of the DNA which is introduced into the cells depend upon the vector system that is being used. In one fate, the DNA that is introduced in the cell replicate and expresses without integration. That is, the DNA is maintained in the nucleus into the extra chromosomal state. That is, DNA is maintained episomally. If DNA replicate and expresses without integration into the genomic DNA of that particular organism, and it is maintained in nucleus into the extra chromosomal state, then that is known as episomal. And this kind of transfection is known as transient infection. It is known as transient transfection. What is transient transfection? In this phenomenon, the DNA which is introduced in the cells, it does not integrate with the chromosome of that particular cell. It is maintained in nucleus in the extra chromosomal state, and that is how it replicate and expresses. This is what the transient transfection is all about. Now, in the second fate, the DNA integrates at the random chromosomal site on the host genome, and it replicates as a normal part of the genome. In the second fate, the DNA is integrating with the chromosome of the host genome, and after integration, that DNA replicates as the normal chromosome replicate. That is the second fate. If the introduced DNA integrates into the host genome and it is maintained permanently in the cell, then that kind of transfection is known as stable transfection. Okay, so we have two type of transfection. One, we had transient transfection, and this is stable transfection. In transient transfection, the foreign DNA is maintained in an extra chromosomal state, and in this stable transfection, the foreign DNA is integrated into the host genome and it is maintained permanently in the cell. If the transfected exogenous DNA is non-replicative, then stable transfection must occur by integration of DNA into the genome. Okay, if the exogenous DNA that you are inserting into a particular animal cell, if that DNA is able to replicate on its own, then there is no problem. But if that exogenous DNA is non-replicative, then stable transformation must occur and there should be integration of DNA into the genome for the sake of replication and expression. Now let's discuss about some chemical transfection strategies. Now, principle of chemical transfection involves the interaction between negatively charged nucleic acid and positively charged carrier molecules such as polymers and lipids. Okay, in chemical transfection there is interaction between negatively charged nucleic acid and positively charged carrier molecules, and this interaction enables the nucleic acid to come in contact with the negatively charged membrane proteins, and that leads to incorporation of the gene into cell by endocytosis. Okay, so there is interaction between the nucleic acid and carrier molecule. That interaction leads the nucleic acid to be in contact with the negatively charged membrane proteins, and this leads to incorporation of the gene into cell by endocytosis. Okay, that is how your chemical transfection occurs. Now let's see transfection with calcium phosphate. Now this transfection method is done by washing the cultured cell with phosphate buffer and to it adding DNA and calcium chloride. You take phosphate buffer, you add your cultured cell into it, and then you add DNA and then calcium chloride. After that, what happens into that mixture is the precipitate settles on the surface of cell, and that precipitate is then internalized by endocytosis. Okay, that precipitate has the DNA, and when DNA precipitates settles on the surface of cell, those precipitates are internalized by endocytosis, and that is how your DNA is introduced into your cultured cell. Okay, so that's all about the transfection with calcium phosphate. Now let's look at the transfection with DEAE dextran. Now this chemical transfection method utilizes DEAE dextran, that is diethyl amino ethyl dextran, and this is a soluble polycationic carbohydrate, and this DEAE promotes the interaction between the DNA and cell, and thus it promotes the internalization of the DNA into cell. Okay, this diethyl amino ethyl dextran, this DEAE dextran, is a soluble polycationic carbohydrate, and it promotes the interaction between the DNA and cell, and thus it promotes the internalization of the DNA into the cell. Now let's discuss about liposomes and lipofection. Now liposomes are vesicles that have aqueous compartment that is enclosed by a phospholipid bilayer. Okay, liposomes are the vesicles which have an aqueous compartment enclosed by phospholipid bilayer. Now these liposomes can act as a DNA delivery system by two ways. Either you can entrap the DNA into the aqueous compartment of these liposomes, or you can complex the DNA on the phospholipid bilayer of the liposomes, and then you treat the liposome which have the DNA with the cell, and the DNA of the liposome will go into the cell, and that is how this liposome method is going to work out. Okay, so when you mix this liposome with the cells of culture, then this liposome vesicles fuse with the cell membrane of these cells and deliver the DNA into the cytoplasm. Okay, when you mix liposome with the cell of the culture, those cells into which you want to introduce your foreign gene, then the vesicles, okay, these liposomes fuse with the cell membranes of these cells and deliver the DNA into the cytoplasm. Now, the efficiency of this liposome mediated gene transfer can be enhanced by incorporating viral proteins. Okay, you can incorporate viral proteins, and that is how you can enhance the efficiency of this liposome mediated gene transfer. Okay, what does viral protein do? Why do you incorporate them? Okay, these viral proteins facilitate the active fusion between the viral envelope and the cell membrane. Okay, what is the natural function of these viral proteins? They facilitate the active fusion between the viral envelope and the cell membrane, and that is the reason why you can incorporate the viral proteins into the system, and that they will enhance the efficiency of the liposome mediated gene transfer. Okay, so after the process, the fusogenic particles which are produced are known as virosomes. Okay, virosomes are those liposomes which have viral proteins in them that promotes the fusion process. Now, lipofection involves cationic or neutral lipid mixtures which spontaneously associated with negatively charged DNA to form complexes. Okay, there is this process lipofection. What does it do? Okay, it involves cationic lipid mixture or neutral lipid mixture, and this thing spontaneously associate with negatively charged DNA, and that forms complexes. Now, in liposome mediated transfection, the DNA is encapsulated into the lipid vesicle. Okay, this is what is happening in liposome mediated transfection. But in lipofection, what is happening? There is formation of the DNA lipid complex, which is known as lipoplex, and this DNA lipid complex is then taken up by the particular cell by endocytosis. Okay, so in liposome mediated transfection, you put the DNA into the lipid, and then that lipid fuse with the cell membrane of the target cell and then introduce that DNA into the cytoplasm. Okay, that is what is happening in liposome mediated transfection. But in case of lipofection, there is formation of this DNA lipid complex, which is also known as lipoplex, and after this complex has formed, when you treat this complex with the cells, then cells take up this DNA lipid complex by endocytosis, and that is how you introduce the DNA into your target cell by lipofection. Now let's discuss about cell or protoplast fusion. Now there are certain chemicals which are known as fusogens, such as polyethylene glycol, which is abbreviated as PEG, and these chemicals cause the cell membrane to fuse. Okay, in the presence of these fusogens like polyethylene glycol, the cell membranes fuse. Okay, so this phenomenon can be exploited to transfect the animal cells by mixing the animal cells with other cells that has large amount of plasmid DNA. Okay, you can prepare a mixture in which there are animal cells and there are other cells which have large amount of plasmid DNA, and you can introduce into that mixture these fusogens, and that will lead to the fusion of the membrane of animal cell with these other cells, and that is how the plasmid DNA will be introduced into the animal cell. Now this scientist Schaffner first used bacterial protoplast to transfect the mammalian cell. Okay, he used bacterial protoplast to transfect the mammalian cell by treating the bacterial cell with chloramphenicol and lysozyme. He used chloramphenicol to amplify the plasmic content, and lysozyme to remove the cell wall. Okay, so this scientist first used the bacterial protoplast to transfect the mammalian cells. So he treated bacterial cell with this chloramphenicol and lysozyme, and then the protoplast of this bacterial cell was induced to fuse with mammalian cell, and that is how the process went. Now let's discuss about receptor mediated transfection. Now this receptor mediated transfection involves the delivery of DNA to a particular cell by conjugation to a specific ligand. There is conjugation to a specific ligand, and that is how the delivery of DNA happens to a particular cell in receptor mediated transfection. Okay, the ligand interact with the receptor on the cell surface, and this interaction allows the ligand and the attached DNA to be internalized. Okay, ligand interact with the receptor of the cell surface, and this interaction then allows the internalization of the attached DNA into the cell. Now, one problem associated with this receptor mediated transfection is that the ligand DNA complexes are internalized as endocytotic vesicles. Okay, the ligand DNA complexes are internalized as endocytotic vesicles, which then fuse with lysozyme, and if the vesicle is fusing with lysozymes, then there will be degradation of the DNA, and this will lead to the failure of the gene expression. Okay, so the vesicle that is internalized into the cell, that goes and fuse with the lysosomes, and then there is degradation of DNA, and this is a problem associated with the receptor mediated transfection. But the thing is that some DNA escapes this fate and it goes into the nucleus and it gets expressed there, and the mechanism of this is not understood. But yeah, some DNA of that endocytotic vesicle gets into the nucleus and it is expressed there. Okay, this receptor mediated transfection is highly efficient in cell cultures, and it results into the transfection of 90% of the cells which have appropriate receptor. Okay, it is a highly efficient method. Okay, this receptor mediated transfection results into the transfection of 90% cells which carry appropriate receptors. Now, less success has been observed in case of in vivo gene transfer because ligand DNA complexes are degraded in serum, and the thing is that size of particles serve as a critical parameter for the transfection of different cell types. Okay, so there is less success in vivo gene transfer by using this receptor mediated transfection strategy. Okay, so that's all for this video. In the next video, we are going to discuss about physical transfection strategies and transduction as well. So see you next time. Thanks for watching.